Assalamualaikum and hi everyone. Today our group would like to show you how to do experiment on DNA extraction.
The first thing is we must have the E. coli suspension. The E. coli suspension are transferred into 1.5 ml of microcentrifuge tube. After E. coli has transferred into the microcentrifuge tube, these tubes need to be centrifuged in the centrifuge for 12,000 G for about 1 minute. Now we have to wait for about 1 minute. After 1 minute, the centrifuge are stopped and we can take out our microcentrifuge tube. Now we can see that we have a supernatant inside this, on the top of this tube, and the supernatant need to be discarded because we only want the cell on the bottom of the tube. Let's discard the supernatant. For the next step, we need to have lysis buffer and also micropipette. The micropipette are used to transfer the lysis buffer into the tube. Now, we need to adjust first the micropipette to 100 microliter because we want to transfer 100 microliter of lysis buffer into the tube. Now, let's pipette the buffer. Make sure that we have 100 microliter of lysis buffer and are transferred into the tube containing our cell. Make sure to press the micropipette for 2 times until the last stop so that all our lysis buffer are transferred fully into the tube. For the third step, we need the usage of micropipette and also proteinase K. Firstly, don't forget to adjust your micropipette into 20 microliter because we want to transfer 20 microliter of proteinase K into the tube. Let's take 20 microliter of proteinase K and transfer it into the tube containing our cell and also lysis buffer that we had put on the previous step. Don't forget to push until the last stop of the micropipette.
The tube was incubated at 55 degrees Celsius for 15 minutes. Next, the tube was flicked every 5 minutes to make sure the mixing is well mixed. 20 microliter RNase A was pipetted out and added into the sample and mixed. RNase A is functioned to degrade protein. Then, incubate the mixture for 2 minutes at room temperature to degrade the RNA. Next, 500 microliter binding wafer was pipetted out and added to the mixture. The mixture then vortex for 30 seconds.
The entire contents were transferred to a spin column, then centrifuged at 12,000 RPM for 30 seconds. After 30 seconds, the flow was discarded into the discard bin. After that, 500 microliter of CB211 buffer was added and centrifuged at 12,000 RPM for 30 seconds. After 30 seconds, the flow was discarded into the discard bin. Then, 500 microliter of washing buffer WB11 was added and 96% to 100% of ethanol had been added. After that, centrifuge at 12,000 rpm for 30 seconds. After 30 seconds, the fluid was discarded into the discard bin. After that, the step was repeated once. Add 500 microliter of washing buffer WB11 and 96% to 100% of ethanol has been added and centrifuge at 12,000 rpm for 30 seconds. Again, after 30 seconds, the fluid was discarded into the discard bin.
Centrifuge the empty column at the maximum speed, which is more than 12,000 xg, for about 2 minutes. Next, place the spin column in a sterile 1.5 ml microcentrifuge tube. Then, pipette 200 microliter of binding buffer that has been preheated to 65 degrees Celsius into the column matrix and incubate the column matrix at room temperature for 2 minutes. Then, centrifuge the column matrix at 12,000 xg for 1 minute to elute the isolated genomic DNA. Store the isolated DNA at -20 degrees Celsius.